Next, I'm going to read a little bit from Chapter 8, Microbial Metabolisms. I'm going to first talk about energy cells. Energy cells have exergonic reactions, which release energy as they go forward, and energy is also available for doing cellular work. Endergonic reactions require the addition of energy to move forward. Exergonic and endogenic reactions are often coupled, proceed in the same space and time. ATP synthesis is an endogenic reaction coupled to reactions of cellular respiration, oxidation of glucose. And here is a simplified model of energy production, burning glucose. It shows you how glucose oxidizes and turns into an enzyme catalyzed pathway, which produces your end products. And this oxidation releases hydrogen ions with electrons, and it turns into ATP. Just like you have to make money to make complex things, like life, things happen. Bacteria do the exact same thing. Bacteria constantly release energy through breakdown of complex things. That energy is captured in a form of ATP, which is a reaction of degradation is coupled to reaction of ATP. Now we'll talk about adenosine triphosphate, energy conservation, money of the cell. There are three parts of the molecule, which is the adenine, which is a nitrogen base, a ribose, which is a 5-carbon sugar, and 3 phosphate groups bonded to the ribosome. Bulky and have a negative charge, repelling electrostatic charges impose a strain between the last 2 phosphate groups, and removal of phosphate energy releases free energy. Now, I'm going to talk about ATP and substrate level phosphorylation. This is a primary energy currency of the cell. When it is used in a chemical reaction, it must be replaced. ATP utilization and replenishment is an ongoing cycle. 3 ways of ATP synthesis is a substrate level phosin, oxidative phosphorylation, and phosphorylation. Substrate level phosphorylation is a very efficient way, but it works. It has a very low rate. Generation of ATP through a transfer of a phosphate group from a phosphorylated compound directly to ADP does not yield too many ATPs. So you might be wondering, where does the cell use it? It is used in glycose and fermentation and in citric acid cycle, and electron carriers and hydrogen ion gradient are not required in this cycle. There are several other ATP synthesis pathways, and they require a membrane and an electron carrier and 3 generation of proton motive force. A series of redox reactions occurring during the final phase of the cellular respiration pathway called electron transfer chain or electron transport system. These yield a lot more ATPs than substrate level PHSNs. Where do cells use it is when it's used with cellular respiration, also known as ETC. Photophosphorylation is similar to oxidative, but light is involved. ATP formed through a series of sunlight-driven reactions in phototrophs, which is during the light phase of photosynthesis, is similar in ETC, which is the cellular respiration. Electron carriers are involved as in oxidative phosphorylation. And this yields a lot more ATP molecules than substrate level PHSNs. There are 3 known basic catabolic pathways, and that's aerobic respiration, anaerobic respiration, and fermentation. And glycolysis is the most common pathway used to break down glucose. There are several elements of electron transport, which is such as ATP synthase and oxidative phosphorylation, which is chemiosmosis, and proton motive. Proton motive is the coupling of ATP synthesis to electron transport. Each NADH that enters the ETS gives rise to 3 ATP molecules. and H plus can only diffuse through the membrane.